Hello students, today there is my second lecture of acquired immunity or adaptive immunity. In first lecture, we already have discussed about antigen presenting cells. So now T cell and B cell, that is CD antigen of these cells. So in acquired immunity or adaptive immunity, as we know, the adaptive immune or specific immune response consists of antibody responses and cell mediated responses which are carried out by different lymphocyte cells, B cells and T cells respectively. B cells are the major cells involved in the creations of antibodies, that is humoral immunity, that circulate in blood plasma and lymph, where they bind specifically to foreign antigens. And in cell-mediated immunity does not involve antibodies but rather involves the activation of macrophages, natural killer cells, that is NK cells, antigen-specific cytotoxic T lymphocytes, and the release of various cytokines in response to an antigen. When immunologists began to study different kinds of lymphocytes, they began to classify them by CD antigens that were on their surface as known as CD4 plus T cells are helper T cells and CD8 plus T cells are cytotoxic T cells. So these CD antigens for cluster of differentiation, CD, that is cluster of differentiation, which indicates a defined subset of cellular surface receptor, that is epitopes, that identifies cell type and stage of differentiation and which are recognized by antibodies. Actually जब ये स्टडी की जा रही थी इम्यून सिस्टम की, तो साइंटिस्ट को बहुत ज़्यादा क्लस्टर्स में टी सेल और बी सेल्स के क्लस्टर्स में एक डिफ्रेंशिएशन मिल रहे थे, तो एक उसको आइडेंटिफिकेशन के लिए, डिस्प्ले के लिए ये नेम दे दिए गए. लॉट्स ऑफ मॉलिक्यूल प्रेजेंट ऑन द सरफेस. आई थिंक more than 200 CD clusters of differentiation molecules are known. So CD antigens, they may present on the T cell or they may present on the B cells and other cells also. So here there is a T cell CD antigens. CD4 is as membrane glycoprotein expressed predominantly on T cell, providing the surface protein to which HIV attaches itself in order to invade the cell. HIV is example of foreign particles. CD4 is also found on the surface of monocytes, macrophages, dendritic cells, astrocytes, keratinocytes and glial cells. CD8 is a co-receptor for the T cells receptor TCR. CD8 is also be found on natural killer cell, cortical thymocytes and dendritic cells. Another type of CD antigen, that is CD58, is also known as lymphocyte function associated antigen, is also present on the T cells, that is known as LFA3. Now CD antigen was present on the B cell. CD40 is belonging to the TNF, that is tumor necrosis factor receptors superfamily. So CD40 is belonging to the TNF receptor superfamily. ये ट्यूमर नेक्रोसिस फैक्टर के जो रेसेप्टर सुपर फैमिली है उसमें इंक्लूड होने वाला ये एंटीजन होगा. तो CD40 is expressed on B cells, dendritic cells, activated monocyte, macrophages, endothelial cells, vascular smooth muscle cells and several tumor cells lines. Human and mouse CD40s have 64% of identity of amino acid sequence identity. CD72 is a membrane glycoprotein of the C-type lectin family. CD72 is a pan-B cell marker that is expressed throughout the B lymphocyte differentiation except plasma cells. And CD72 is also present on the follicular dendritic cells. Some other CD antigens which are present on the B cells, that are CD10, CD19, CD20, CD22, 35, 37, etc. So, number of CD antigens which are present on the T cell or B cell or other cells. आप अगर फॉरेन बुक्स की एंड में जाके देखिए, अगर कुबे की बुक के एंड में जाइए, आपको मोर देन 225 जो है वो CD एंटीजेंस के नंबरिंग मिलेगी और दे हैव different types of functions. Now here अभी हम लोगों ने ब्रीफ में देखा कि CD4 as a helper T cell and CD8 is known as cytotoxic T cells. So here there is a is the CD4 and B, that is CD8 bearing T cells. It means there is a presence of T cell having the glycoprotein, a protein molecule on their surface, that is 4 kinds of. ठीक है, 4 numbering that is depends on the discovery, 1 2 3 4 5 6 7 8. So there is a CD4 which present on the T cell. This one is the T cell. Okay, तो ये हमारा helper T cell, that is. So there is a activation. How T cell activated? That are 2 kinds of T cell, helper T cell and cytotoxic T cell. So in case of A, figure A shows that CD4 is associated with the helper and regulatory T cell. Okay, helper is a regulatory T cells. And extracellular pathogen ये देखिए आपका एक्स्ट्रासेलुलर पैथोजन जो हमने लास्ट टर्न पे एंटीजन प्रेजेंटिंग सेल के लिए पढ़ा था कि जो एक्स्ट्रासेलुलर पैथोजन से लाइक बैक्टीरिया is processed and presented in the binding cleft of a class 2 MHC. There is the class 2 MHC. Here pathogen or antigen is held by the MHC class 2 molecule, and this interaction is strengthened by the CD4 molecule. Actually यहाँ पे जो CD4 molecule है वो strengthen करता है हमारे इस interactions को. एक तो यह है जो हमारी जो T cell है उसका this receptor, T cell receptor, look here, this is the T cell receptor, TCR जिसको हम कहते हैं TCR. Okay, so antigen is recognized by TCR and CD4 molecule which confirm the MHC, presence of MHC. Actually CD4 जो है वो confirm करता है कि antigen is presented by MHC or not. अगर ये MHC जो है एंटीजन को होल्ड नहीं कर रहा है, प्रेजेंट नहीं कर रहा है, अगर एंटीजन फ्री है, सो देयर हेल्पर टी सेल डज नॉट इंटरटेन द केस. वो जो है फिर एंटीजन को रिकग्नाइज नहीं करेगा, वो इंटरैक्ट नहीं करेगा. ये हमेशा जो है वो होगा कि विदाउट एमएचसी जो एंटीजन्स होंगे, एक्सप्रेस होंगे या इंटरैक्ट करेंगे, उसे जो है टी हेल्पर सेल इंटरैक्ट नहीं करेगा, उसको एक तरह से रिस्पॉन्स नहीं करेगा, रिकग्नाइज नहीं करेगा. तो इट इज़ कंपल्सरी दैट कि जो एंटीजन है उसको प्रेजेंट करने वाला एमएचसी वहाँ प्रेजेंट हो. सो ये है द एमएचसी क्लास 2. एंड व्हेन इट इज़ एक्टिवेटेड, व्हेन एमएचसी विथ एंटीजन recognized by the T cell, then there is a activation of T cell, and activated T cells release cytokines, number of protein molecule secreted by T cells, and these cytokines T cells destroy infected cells by releasing enzyme that rupture सेल मेम्ब्रेन। फिर जो भी हमारा जो पैथोजन होगा उसके ऊपर ये जो है एंजाइम सिक्रीट करेंगे और उसको डिस्ट्रॉय करेंगे। इन सेकेंड केस वेयर पैथोजन इज intracellular. So an intracellular pathogen is presented by a cell MHC class 1 present the antigenic domains or antigen by a class 1 MHC molecule and CD8 interact with it. यहाँ पे जो देखिए यहाँ CD8 interact with the molecule. तो ये एक तरह से ये हमारा टी हेल्प रिसेप्टर हो गया, टी सेल रिसेप्टर जो साइटोटॉक्सिक टी सेल है. देयर इज़ अ टी सेल साइटो एक्टिवेटेड टी सी सेल और साइटोटॉक्सिक टी सेल activated in the presence of antigen which is expressed by the MHC class 1, and after the activation these T cells proliferate, number of clones are present and secrete the granzyme that is the enzymes, and this enzyme and perforins destroy the pathogens. Okay, so there is a T helper cell and CD helper cell. Now here in this diagram how cytotoxic T cell work. So look here, there is a naive T cell. Naive T cell, it is in fact immature. When this naive T cell interact with the MHC class 1 with antigen, as you know, antigen presenting cell, that is the presented antigen by MHC class 1. So there is a activation of TC. Okay, so here antigen extracted from a pathogen, ये यहाँ से एंटीजन ये आपका कांटेक्ट में आ गया. तो receptors which present on the TC which recognize the antigen and CD8 which also help in the interaction confirm the presence of MHC, and activation of this, there is a function in two way. Activated TC cell, cytotoxic T cells, are cloned and destroy infected cells in the body. अब जब ये एक्टिवेशन हो गया तो TC सेल का जो है वो मल्टीप्लिकेशन होगा, माइटोसिस होगी और वो नंबर ऑफ क्लोन्स फॉर्म होंगे एंड दीज क्लोन्स डिस्ट्रॉय द पैथोजन. ओके, एंड इन अनदर वे देयर इज़ अ फॉर मेमोरी सेल, मेमोरी टी सेल. एडॉप्टिव इम्यूनिटी के लिए हमेशा ध्यान में रखना है, if memory cell is formed then it go into adaptive immunity or acquired immunity. These memory cell TC cells are produced and are activated if this antigen is encountered again. memory cell का क्या काम है, जब हमारा इस पैथोजन का दोबारा इन्फेक्शन होगा तो हमारे बॉडी में जो मेमोरी सेल प्रेजेंट हैं इन द फॉर्म ऑफ TC सेल, वो जो है शॉर्ट ड्यूरेशन में जो है वो क्विक रिस्पॉन्सेस करेंगे. मतलब उनके लिए रिस्पॉन्स करने में बहुत ज़्यादा टाइम नहीं लगेगा जितना फर्स्ट रिस्पॉन्स में दिया गया. तो वो मेमोरी सेल पैथोजन को रिकॉग्नाइज कर लेंगी और उसे किल करने में एक शॉर्ट ड्यूरेशन में ही वो एक्टिवेट होके अपनी प्रोसेसेस स्टार्ट कर देंगी और डिस्ट्रॉय करेंगी पैथोजन के. ओके, तो इस तरह से हम लोगों ने क्या देखा, देयर इज़ अ CD antigen that is CD4 plus and CD8 plus it means the presence of this glycoprotein or protein on the surface, kind of 4 or kind of 8, that is cell, C for cell, not C for cluster and D for differentiation. So cluster of differentiation, that is a short name. So CD4 cell which present on T helper cell and CD8 that is on CD cytotoxic T cell. Is it clear कि हमेशा जो 4 है CD4 है वो हेल्पर T सेल में मिलेगा आपको। अब हम इसके आगे फिर देखेंगे कि ये हेल्पर टी सेल के सबसेट्स क्या हैं, टी हेल्पर 1, टी हेल्पर 2। हम नेक्स्ट लेक्चर में इसको अब देखते हैं कि व्हाट इज़ द सबसेट्स ऑफ टी हेल्पर सेल्स। एंड साइटोटॉक्सिक टी सेल दैट इज हैविंग अ kind of glycoprotein on their surface, that is CD8. ठीक है, and CD8 which recognize antigen presented by the MHC class 1, MHC class 1, and this MHC CD4 that recognize the T helper cell, T helper cell having CD4 recognize the antigen that is presented by MHC class 2 molecule. Okay, so this is the brief. So T cell and B cells जिस पे CD है, हमें जो है मेन यहाँ पे देखना है कि अभी हम T सेल पे जो प्रेजेंट हमारे क्लस्टर्स हैं, जो ग्लाइकोप्रोटीन है सरफेस पे, दैट इज CD4+ and CD8. Okay, is it clear? Thank you.